Hi, Alex. How are you?
I am doing great, ma'am. Thanks for asking.
Well, Alex, how is the exam preparations going?
Well, I am stuck in a single topic. The terms inside are confusing me big time.
Can you tell me which topic?
It's resting membrane potential. Can you help me with this topic? Please?
Turns out, Alex, that it's your lucky day. I am delivering a lecture on resting membrane potential in 30 minutes. Do you have time to attend that?
Yeah. My classes are over for the day. So I can join you.
Perfect. See you there.
Thanks, ma'am.
Hi, students. We have Alex here today with us. He will also be learning about today's topic, which is resting membrane potential.
Welcome, Alex.
So let us start. Before diving deep into resting membrane potential, we need to understand that what is membrane potential anyway. Membrane potential in simple words is the potential difference across the cell membrane. Potential difference meaning the difference in charges across the cell membrane, which can be either positive or negative. If we look at the structure of cell membrane, it is basically lipid bilayer that protects the cell from outside environment. This is a typical diagram of cell membrane. Now let us mark this side as outer environment or extracellular space. And this will be inner environment or intracellular space. Now, how does this membrane potential is created? Well, my students, there are several ions inside and outside the cells. Their concentration decides that either the overall charge on the cell is negative or the overall charge on the cell is positive. There are several ions in our body. Let's say they are sodium, potassium, chloride, and calcium ions. Now imagine that you have 6 sodium ions outside the cell and 4 potassium ions inside the cell. Which side do you think will be more positive?
I think 6 sodium makes plus 6 charge, so outside the cell will be more positive.
That's right, Alex. The outside will be more positive. And the inside of the cell will have plus 4 charge of potassium, so the inside of the cell will be more negative. This difference in concentrations of ions is known as the potential difference across the cell membrane.
So, is this called membrane potential?
That's right. This is a typical example of membrane potential. Potential difference is also known as voltage difference or ion concentration difference, so don't get confused by different literature books. Now the membrane potential, which is the most common one, is the resting membrane potential. Resting membrane potential is the potential difference when the cell is at rest or non-excited state.
Does that mean that there will be a potential difference in the cells at the excited state as well?
Yes, but that is known as an action potential. It is created when the cells are in an excited state.
So, does resting membrane potential occur in all cells?
Yes, resting membrane potential occurs in all cells. However, action potential does not occur in all cells.
And why is that?
Alex, this is because not all cells have the capability of producing electric current or nerve signals. This can occur in cells like neurons and muscle cells.
Oh, that's the important thing. Let me write that down.
Sure. While you are writing this down, also note that the resting membrane potential always has a negative charge on the inside of the cell and a positive charge on the outside of the cell.
And what is the reason for that?
This is because of the need for cells to create an electrochemical gradient. The electrochemical gradient makes a difference in charges inside and outside a cell.
So, what happens if there is no electrochemical gradient?
Very good question, Alex. Let me tell you this with an example. Suppose you have equal charges outside and inside the cell. You have 5 sodium on the outside and 5 sodium on the inside. What will be the net charge?
The net charge will be 0.
Yes, you are right. So now that the charge is 0 and everything is balanced on both sides, how will I perform cellular functions like nerve impulse generation or muscle movement?
Oh, you are right. So, in order for a cell to perform its functions, we need an electrochemical gradient.
Yes. And why negative charge on the inside? This is because the positive ion concentration is limited inside the cell. Less positive inside, more positive outside.
This makes a lot of sense now.
Let us take an example of nerve fiber and study its resting membrane potential. Now, you need to remember that there are 2 channels responsible for maintaining resting membrane potential. The most important one is the sodium-potassium ATPase pump, and the second one is the potassium leak channels. There is a pump located inside the cell known as the sodium-potassium pump. This pump is responsible for moving sodium and potassium ions inside or outside of the cell. How does this movement occur? This movement will occur due to the breakdown of ATP. ATP breaks down and forms ADP and inorganic phosphate with the help of the enzyme ATPase. This breakdown releases energy that drives the movement of ions outside and inside the cell. This pump has a principle of moving 3 sodium outside the cell and 2 potassium inside the cell. Can anyone tell me what happens to the charge of the cell?
I think I can tell. If 3 sodium ions are moving outside and 2 potassium ions are moving inside, then there will be more positive charge on the outside and less positive charge on the inside. Right?
This is so right. This movement of charges will create a large electrochemical gradient for sodium and potassium across the resting membrane potential. There are specified values of sodium and potassium outside and inside the cell. Sodium ions outside the cell are 142 milliequivalents per liter. And sodium ions inside the cell are 14 milliequivalents per liter. While potassium ions outside the cell are 4 milliequivalents per liter. And potassium ions inside the cell are 140 milliequivalents per liter. Alex, you might want to note these down for your exams.
Way ahead of you, ma'am. Please tell me about the other channel as well, which is the potassium leak channel.
Certainly, in the nerve cells, potassium ions can leak through nerve membranes even in resting cells. This leakage is also responsible for the overall negative charge inside the cell membrane. Sodium might also leak from the channels, but potassium leakage is around 100 times more than sodium ions. The net resting membrane potential inside the cell is about -70 millivolts.
And why are we saying that this value is negative?
This is because the inside of the cell is negatively charged. The negative charge represents that there is negativity inside the membrane, and in order to minimize this charge, we need to move positive charges inside the cell. This is where action potential will kick in. But that is the topic for another day.
This was really interesting. Can you also teach us how to calculate this potential difference?
Yes, let us calculate how we have achieved this -70 millivolts of resting membrane potential. Let us move toward the next section.
Discover a wealth of accurate, up-to-date medical videos for in-depth learning, anytime, anywhere. Elevate your medical expertise with sqadia.com.